Our next presentation is from Sam Doesburg. In some respects, this talk and my talk this fall is kind of practice for a course that he and I are teaching this semester. Although we're not going to do it concurrently, which would be difficult for the students, of course. I don't think you can handle both of us in the same day. But Sam is a fellow faculty member at the Department of Biomedical Physiology and Kinesiology, or BPK. He's also been doing a lot of work in MEG and development, so that's why he's actually a good person to be giving this talk. He was in Toronto for a number of years. That's where I first met him, and then came out to SFU 2015. So, and he's going to tell us about some of the machines that are used to measure brain dynamics. And then I will introduce myself later awkwardly and talk about the modeling aspect. Sam, please.
Okay. People on Zoom, let me know if you can't hear me through the lapel mic. So hopefully this isn't all too remedial for you guys, but my job is just to get everybody up to speed on fMRI and EEG and everything, just to make sure that everybody understands what comes next. So I'm just going to give a general overview of the methods. I'm going to talk a lot about the structural imaging, including a focus on diffusion tensor imaging, because that's the most common modality to infer non-invasively the structural brain connectivity. And then I'm going to talk about the various modalities that we use to try to image the function and dynamics and connectivity of the brain. So EEG, MEG, intracranial EEG, which has already been touched on a lot, and fMRI, which has been very prolific. And importantly, to understand how each of these sort of enables us, but how each of these also sort of constrains us, because all of these have their advantages, but they all have their unique limitations, their unique disadvantages. So whenever we're looking at data, it's always very important to ask ourselves, what does this tell us, but also what does this not tell us? How does it bias? So I like to start and end these kinds of talks with this type of slide, because this kind of places each of the common modalities with respect to their strengths and weaknesses as we look for the balance between the trade-off between temporal resolution, timing accuracy, and spatial resolution. So there is no perfect, we like to fight who's got the better machine, but there is no perfect one. We see an extreme example, the ever popular scalp EEG, because it gives you a direct electrophysiological measure, and it does that with millisecond time precision, but it is extremely poor in spatial resolution, because the electrical fields are sort of smeared out as they pass through the skull and meninges, but it's compared to MRIs, very cheap, very portable, fairly simple. MEG is a little bit with spatial resolution, but gives up a lot of that portability, a lot of that simplicity, a lot of that low cost that comes with scalp EEG. fMRI has probably been the most prolific in terms of contributing to our understanding of brain connectivity, brain dynamics, and that's because the machines are great. They give us all kinds of information about structure as well as function. They give us high resolution functional mapping, non-invasively throughout brain space. And they don't have some of the limitations that EEG and MEG do, like it works for deep sources, it works well for sources irrespective of the orientation of the neurons, the pyramidal cells. But the disadvantages of fMRI are that it's quite slow in terms of its acquisition rate, and it's an indirect measure. So you're always looking at blood oxygenation level dependent signal, which is not really recorded from the neurons themselves, but it's inferring from, that through a metabolic measure. And you see sort of FNIRS in the middle, which has issues with depth. So we're talking a lot about networks. So it's important to introduce this idea of graph theory or graph analysis. It's not new, but I think it's fair to say that it's made a huge splash in the neurosciences in this. If you look at the history of neuroscience in a simplistic way as a tug of war between localizationism, ascribing particular function to a particular brain area, particular cortical column, and connectivity, describing function as arising from interactions between different elements. The introduction of graph analysis in the context of neuroimaging and other recording techniques has created sort of a connectionist renaissance in the neurosciences. And the way that graph analysis tends to work is to conceive of everything in a network where you have to define your nodes, which are in neuroscience parlance probably your brain regions, as in like a Brodmann area type entity in terms of size, all the way down to a particular implanted electrode, which could be cortical column size, and then edges, which are connections between the nodes. So in a structural network, you'd parcellate, use something like diffusion tensor imaging to define the connections between various areas. And then you'd create a graph. So each row and column is a node, and each sort of color represents the coupling strength between that. Or in function, we'd have implanted electrodes like the stereotaxic intracranial EEG you saw before, reconstructed sources from MEG, et cetera. And then from that, we can extract metrics, which can either quantify the participation of a given node in the network, such as the clustering coefficient, sort of like a local neighborhood index. So if A is connected to B and B is connected to C, what are the odds that B and C are connected? Path lengths or efficiency, or centrality, or a measure of hubness, right? How if you're going from any point A to any point B through the network, what are the chances that you have to pass through a given node to get from A to B? And then there are also metrics which characterize not a particular node, think brain region, but the network as a whole, like small worldness or global efficiency. And a lot has been done by simply using imaging to extract these graphs and graph theory to sort of explain the properties. So you can look at this as a sort of convergence between localization and connectionism in a sense, where we've always attempted to describe particular Brodmann areas, particular functions, primary visual cortex, et cetera, particular columns, particular function orientation columns, et cetera, so that what the sort of current understanding that dominates the field now is I think that we think about the structure of the brain as something that is relatively invariant or stable, but enables and constrains the sort of dynamic repertoire of functional connectivity, which can change much more quickly and represents the sort of dynamics of mental life, cognitive abilities, perception, et cetera. Okay, back to the basics. So the very old-fashioned way of looking at an MRI is to just take an anatomical T1 or T2 image. So this is the kind of thing that you might get if a neurologist suspected there was something wrong with your brain, and it's something that we would acquire in the scanner to get basically a representation of your individual brain anatomy. And the way that that would be used clinically would be they'd look for a lesion, something that was atypical just by trained radiologists doing visual inspection. And then we can do other things in a quantitative sense using things like deformation matrix. So a deformation matrix is almost like a set of vectors that you can use to say, what would it take to take my particular brain and stretch it into an atlas template or an average of all of yours? And then we can use that information to ask questions, is the prefrontal cortex larger or smaller in people who have been recently diagnosed with schizophrenia than a control group, et cetera. And this is the important one when we're talking about networks, diffusion tensor imaging. So what diffusion tensor imaging is it takes every voxel, which is a three-dimensional pixel in the brain, and it basically measures the directionality of restriction of water diffusion in that voxel or three-dimensional pixel. So what that means is if you had something like this, a water bottle, water can diffuse quite easily down this axis, but not very easily on this axis or this axis. So the assumption is that when you're in white matter, because you have myelinated groups of axonal projections, that water can sort of flow down the length of the axon, but not across it. So you create a tensor, which indicates the the degree and the direction of water diffusion restriction for each location. And then you use some sort of tractography, like probabilistic tractography that involves some assumptions to essentially connect these up and draw what are the white matter tracts of the brain. So sometimes that's how we get to, okay, this is how we've mapped the structural connectome of a particular person or something like that. So going back to this metaphor of, you can think about it like people always like to use the metaphor of the most recent technological innovation. Like Freud was always talking about steam pressure psychology, right? If you bottle up too much pressure over here, it's going to come exploding out over there in a way that you can't predict. Well, I mean, we're like that with the internet. People think about the brain and its dynamics like the network or a computer network, where you have the physical infrastructure, which enables which information can pass from which location to which other location, and how quickly and how well. And then we have the sort of pattern of communication, which can change constantly depending on the input or the internal dynamics of the communication system. So that's like the function. And the way that people started looking at spontaneous function was probably first through the default mode network, at least in the context of fMRI. So before this, probably the dominant way of looking at the brain was as a sort of black box. Stimulus comes in, brain lights up, the relevant areas become metabolically active, and then a behavior ensues. And the first set of observations that led to the coining of the default mode network was that no matter what task you ask someone to perform, there are certain regions that become less active during task, and they're more active and more correlated when you're sort of off tasks. And these are areas like the precuneus, posterior cingulus cortex, medial prefrontal cortex, et cetera. So there they are in anatomical space, and these traces just represent the sort of correlation in blood oxygenation level dependent signal. So it was also found that if you just take every voxel in the brain and take the fluctuations in blood oxidation level dependent signal from every voxel in the brain, and then you simply ask, are there groups of voxels that sort of activate and deactivate together in an independent component sense of things, then you can pull out the default mode network. And you can also pull out a series of other networks which are anatomically distributed, but functionally related. So you get the executive control network, salience networks, sensory motor areas tend to activate and deactivate together. Same thing with dorsal attention network, et cetera. And this is all sort of very slow, sort of less than 0.1 hertz, typically fluctuations in blood oxygenation level dependent signal. So speaking very coarsely, there's kind of two different types of dynamics that we talk about with fMRI and more generally. So we have the dynamics of that can happen at rest. When you're sitting there without an instruction, brain regions are talking to each other. Those can change over time, but those communication patterns and their transitions are structured. So that's what we'd call resting state connectivity or resting state connectivity dynamics. And then you get the task dependent activation, which is more related to the performance of a specific task. So, like I ask you to remember the location of several objects on a computer screen. And then I compare that to a baseline or a simpler task, and then you get sort of increased activation and increased correlation between the brain areas that subserve the various components of that cognitive task. So in the example that I just gave, sort of visuospatial executive functioning, you get more activation in prefrontal cortex, which is related to executive processes, and more activation in posterior parietal cortex, which is where we have our sort of spatial or even multimodal representation of the world of visual space, and more connections between that. So you can once again, look at this as patterns of communication that are evolving and organizing themselves in a structured way, either spontaneously without task demands or according to the demands of some particular thing that you're trying to do, reading, trying to remember something, etc. So a couple points from this. One of them is very simple. This is sort of methodologically, if you remember that deformation matrix where we kind of warp everybody's brain into the same anatomical atlas space, we do that and that allows us to average and then get group mean average. Another thing that is interesting to note about this, and this is a recurring theme across modalities, is when you look at the stimulation versus the neutral, so that's the task versus the baseline, do something versus do nothing. It's pretty obvious that most of the metabolic activity in the brain is actually related to what we would call background or resting. And even when we're asking somebody to engage in what is a pretty specific cognitive task, that metabolic change is tiny compared to the background. So that's true with fMRI. It's also true with EEG. So a lot of what the brain is doing at any given time is unrelated to what you're doing behaviorally, but it's related to the sort of self-organizing dynamics that are always there. So network connectivity and dynamics. So the place that this shifted now is not just what's connected to what tends to talk, what brain areas tend to talk to which brain other areas, but how does that change, right? So what studies now are often looking at is this idea of dynamic functional connectivity. So if you have somebody in a scanner for a full 10 minutes and you say, give me all of the time series from all of the blood oxidation level dependent signals, and then do something like a temporal independent components analysis, then you can pull out these default mode network, executive network, dorsal attention network, and then we say, okay, fine. So now we have these networks that are present. It's organized communication. It's in the brain. But then when you use a sliding window or you chop it up in some way, we find out that it's not static, which is not really surprising. But not only is it not static, it is dynamic and it's ordered. So you have certain sort of repeating functional connectivity patterns, which will occur. They'll come back again and again and again. And the transition between them is not random. So for example, this study took several states, and you can see if you're in state 2, you're very likely to go back into state 8. And then once you're in state 8, you're quite likely to go back into state 1 or maybe state 6. So that means that left to its own devices, the brain is cycling through ordered states, exploring its dynamic repertoire of functional connectivity states. And it's doing it in a way that is not random, right? There are structured states that recur and the transition between them occurs in an ordered way. I didn't want to go into it too much, but when you go into frequency space, so repeat this experiment with magnetoencephalography and then looking at correlations within particular frequency bands, we can see that a lot of the time, the default mode network seems to be almost like a switching state. And it seems to be mediated by amplitude correlations involving the beta band, about 15 to 30 hertz-ish. So both in time, frequency space and in time, there's an ordered structure to how the brain spontaneously organizes its activity and how it transitions. And then of course, on top of that, once you ask somebody to do something, or a stimulus arrives, here's my taxi, I need to do something, then the external world will impose that its own transition, which is more like what you would call a task demand, like I showed earlier. Okay. Switching gears back to good old fashioned EEG. Great things about EEG. This is an old technology, right? This is 1920s technology. It's relatively very simple. It's relatively very cheap. and it provides a direct measure of neurophysiological activity. So what you're getting here is a potential measured between electrodes or sets of electrodes, and it's generated by the dendritic currents themselves. And a lot of the time what you see is oscillations, especially the alpha oscillation around 10 hertz, which seems to be related to inhibition or idling. So this is Hans Berger, the inventor, and this is when you close your eyes. You get a big burst of alpha activity over the visual cortex, because the visual cortex has gone from an active state, visual interpreting the scene, to an inhibited or relaxed state. So you get tons of alpha when you're in stage 0 sleep, about to pass out, for example, but yet not properly. So this is the basis of EEG. Like I said, the good thing about it is it's a direct neural measure. So if you have the gray matter, which is on the, the outer surface part where the cell bodies of the pyramidal cells are, the main excitatory cells in the cerebral cortex. The nice thing about this is that they tend to be aligned with the same orientation perpendicular to the cortical surface. So when there's a group of these pyramidal cells that are being stimulated, it creates an electrical dipole. And if that electrical dipole is large enough, think many pyramidal cells polarizing or depolarizing at the same time, then it creates a potential which can be viewed, recorded outside the scalp. The downside with this is this volume conduction idea. As it passes through the skull and meninges, it gets smeared out. So it's quite spatially imprecise if the electrode is outside the head. So EEG today is quite splintered. It's old. It's had time to go into lots of different directions. If you go into a typical psychology EEG lab, you might see that something like this: 64, 256 channels, something like that, which enables you to do some basic source solutions. Whereas this potential field coming from requires a lot of assumptions and is probably off still by in the order of centimeters. This thing on the cart is what you would see were you to have a seizure and wind up in a neurological critical care unit. So it's usually just 20 electrodes or so, and it's just a neurologist or a neurology fellow looking for slowing intraictal epileptiform discharges to figure out what's going on. And commercial systems have gone quite radically portable. Like maybe you've seen this Muse system, which is 4 electrodes, doesn't require any gel. You can wear it around. It requires very little expertise or time to apply. Okay. This is the EEG power spectrum. So people talk about them as 1/f plus alpha, which generally means more power at lower frequencies and less power at higher frequencies, with a definitive peak right around alpha, 10 hertz in a healthy adult, which is what Hans Berger's noted. And if you put a digital filter on this, you get activity in different frequency bands. And those frequency bands are known to have different neurophysiological generators in many cases, and also different roles, both in cognition and in pathology. So for example, gamma oscillations, people are often associated that with active cognition, active consciousness. Alpha, on the other hand, is much more closely related to idling or inhibition. So the advantage of EEG is that we're getting much more closer to the actual type of direct population dynamics that are related to cognition, perception, consciousness, and pathology than you get with an fMRI or a PET. So some of these things that you've heard talked about and we'll hear talk about when we're talking about frequency content, things like that, they're pertaining to things that neurologists that are very real and visually observable. So this is on the left, intraictal spike and wave. So that's something you'd see from an epileptic's brain while they were not having a seizure. This is what it would look like as it transitions into a focal seizure, which we talked about, which is hyperexcitation, hypersynchronization. So when you talk about things like dynamics of functional network connectivity, this can be thought about as in a very real sense as a change in power in particular frequencies, and also a change in the synchronization, the timing congruence between those things. and this is slowing. So if you saw this over the left posterior side of a head, you'd say, okay, maybe this person has a tumor. Maybe this person has a previous brain surgery. Maybe this person has a traumatic brain injury. So when we talk about frequency content, we talk about synchronization as connectivity. Sometimes we're talking about these very real, easily observable things that neurologists have been talking about for decades or longer, but not necessarily using that exact language. So this is one way to get around the volume conduction problem. And you heard about it a lot. In the previous talk, there's stereotaxic EEG. So it's insert one essentially wire with 20 or so leads. And the other one, which is getting a little bit less popular, is the subdural EEG grid. So it's placed directly on the brain surface. And there are however many electrodes the epileptologists and surgical team thinks are appropriate. The advantage of this, of course, is that you get rid of the volume conduction problem, more or less. You're recording directly from the brain, but you lose your whole head imaging. You only get the electrodes where the surgeon wants them. And of course, this is never done in a healthy brain because it requires either drilling through the skull or craniotomy. So if you're interested in normal cognition, this always has the deficit that you're talking about a brain that's sufficiently abnormal that it's a candidate for resective neurosurgery. Event-related potentials. I don't know if we're going to talk about them too much in this, but this is essentially just averaging the same type of stimulus and just averaging the voltage. So, boop, 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 beep, boop, 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 beep, boop, and just take the oddballs, average them, and you'll get an event-related potential. So particular negativity that is associated with that. So some of the common ones people talk about, N1, P1, this is recorded from primary auditory cortex, where information from the auditory system first enters the cerebral cortex, primary visual cortex, and is very responsible for encoding the primitive stimulus characteristics. P300, much more frontocentral, related to things like novelty, oddball, transfer of information to consciousness, et cetera, N400, comprehension language, and so forth. Okay, magnetoencephalography, MEG. This is the big attempt to say, can I keep the millisecond temporal accuracy? Can I keep the whole head imaging? Can I keep the whole non-invasive aspect that makes scalp EEG great and get around the problem that the spatial resolution is bad, right? And the reason that's possible is that unlike electrical signals, magnetic fields are essentially transparent to the skulls and meninges. So they don't get distorted when they pass from your brain to the sensors. Bad part about that is up until very recently, the common magnetometers or gradiometers used this are something called SQUIDs, superconducting quantum interference devices, and they require cryogenic environments to operate, which means you've got to sit in this big chair with a giant dewar or thermos full of liquid helium for this thing to function. So goodbye low cost, goodbye simplicity, all that stuff is gone. But what you do get is you get whole head non-invasive neurophysiological imaging. So the principle is the same, right? You get a bunch of pyramidal cells. They're getting an input at the same time. So there's sort of synchronized dendritic currents that creates a dipole. There's in the magnetic field, there's an ingoing and outgoing current according to the right-hand rule. That's picked up by sensors that are arrayed over the surface of the head. This is an example of those sensors, right? So there's a dipolar source there in a sulcus. These little circles are the individual sensors. That's that outgoing and ingoing current. The generator's there. And then you can use some source reconstruction algorithms to infer a model of where that's coming from. And then typically you do that. You take the individual's MRI, you co-register it. So you're putting that person's neuromagnetic activity back on their anatomy. So people love this for research because you have the frequency, you have the time, you have the direct neural measure. People love it for epilepsy surgery because you can get epileptic spikes and you can put them right back on their MRI without having to cut into the head and with having the whole head source solution. And you get time, right? This is the thing that is missing with fMRI, is when you have a person listen to a tone, make a response, perform a task, and you get a time-locked average. Those little black lines are the traces from each of the 100 or 200 magnetic sensors. Then you can look at how that evolves over time. And we can see here from the pattern of the ingoing and outgoing currents that this is probably bilateral auditory cortex. And then we can map how that unfolds in time. So if you're interested in the sequence of activation or the sequence of connectivity or any sort of connectivity dynamics that are happening at rest that's happening at a fast time scale, then MEG is a good option because it is whole head. And when you think about the speed of behavior, the speed of cognition, the speed of perception is not the speed of fMRI recordings. fMRI is much slower. So that's something to keep in mind. All this stuff from fMRI, we're talking about dynamics that are occurring at timescales that are generally slower than the dynamics of your thought, your perceptions, and your behavior. This gets around it, but it has orientation selectivity problems. It's better at things in sulci than things on gyri. This is magnetism, not electricity. EEG is the other way around. And it's not good at things that are deep. So, and it's not as spatially accurate as fMRI. So it has its own baggage. So this is courtesy of Dr. Tony Herdman from UBC. So this is Tony moving his eyes. And there you can see the 151 sensors. And there you can see the projected brain space. So this kind of gives you a sense of the combination of spatial and temporal resolution that's afforded by MEG as a neuroimaging technology, where you can see as you're saccading, you're getting some major visual cortex activation, of course, because you're shifting the visual field, but you're also getting activity in motor cortex, frontal eye fields, things like that, that are controlling the saccades. So this wouldn't be possible with EEG because the spatial resolution is too bad. Wouldn't be possible with fMRI because the temporal resolution is too bad. So you can pull a lot of measures out of an MEG or an EEG. You can do spatial solutions. You can look at the raw magnetic fields. You can convert that into a map of where the alpha is. You can get the frequency spectrum. This is where the power is at what frequency. So this is just your classical Hansberger alpha blocking eyes open versus eyes closed. The alpha power goes down, but if you had somebody with traumatic brain injury, you'd see that that would be shifted lower. And then you could localize that slowing to a particular brain area and say, where is it slowed, right? So it allows this sort of projection of some of these neurophysiological signatures that neurologists have been looking at for a long time into anatomical space. You can get event-related fields like event-related potentials, and importantly, you can get connectivity, right? You can reconstruct activity from throughout the brain space, think Brodmann area level, sort of gross functional anatomy, according to a cognitive neuroscientist, filter it, calculate some measure of connectivity, and then we can talk about the structure of connectivity and the dynamics. And a lot of that is done by looking at oscillations. So we started with oscillations back in 1929 with Hans, and we're still on to oscillations now. And the reason why people are still interested in oscillations now is that it seems to be related to the waxing and waning of inhibition or inhibitory drive in a local cell assembly, which is coupled to the firing rate. So this is not EEG. This is multi-unit activity from an implanted electrode, and this is local field potential from an implanted electrode, sort of the spiking and the low frequency activity. And as you can see, the oscillation, because it's fluctuating in a systematic way, the excitability of the local cortex, it's constraining the spiking in time. And those are the same oscillations that we're seeing at a much coarser scale when we record outside the head. So look at it this way. This became very, I guess, fashionable back in, was it the early 90s as a potential approach to the binding problem. And the binding problem is, you guys all remember Descartes and his convergence zone, the pineal gland. You know, you look at my hand, it's got motion, it's got color, it's got location. And in perception, all of that comes together. And Descartes' take was that all comes together in the pineal gland. More modern take is that there might be nowhere where it completely comes together. So then the solution is when we have integrated percepts, integrated cognitive moments or thoughts, somehow a unique constellation that encodes the various components of that has to be integrated for a given time, and then it has to dissolve to allow for the possibility of a new percept. So you can think about that as the old school switchboard operator trying to create the ideal pattern of communication between the various specialized neuronal elements. And in a very simple way, people started doing this with primary visual cortex. You've all seen this ice cube model of visual cortex. We have cortical columns with particular orientations, and then ocular dominance columns. And people found that when people perceive an object, at a gamma frequency, you see coherence or synchrony between the columns that encode for the different features of my hand in visual cortex. And if it's moving, it also synchronizes with M4 so that it's encoding. The idea is that the oscillations allow for the formation and destruction of transient cell assemblies, which basically encode the contents of momentary consciousness or integrate the neuronal elements required to perform the cognitive task or encode the percept that you're on at the moment. So this is an old idea by Pascal Fraser, but the basic idea is because oscillations are waxing and waning of inhibitory drive, that when you have two, say, cortical columns, or even two large sort of Brodmann area cortical masses oscillating at the right synchrony, they can exchange bursts of action potentials during the depolarized phase of the receiving neuronal groups sort of fluctuations, so they have more downstream impact. So this is a sort of mechanistic account of how it might be possible that neuron group A talks more to neuron group B at time 1, and neuron A talks more to neuron group C at time 2, by essentially changing the coordination of the synchronization between these different neuronal elements at either a columnar level or even up to like a Brodmann area level. So this task-dependent, percept-dependent, action-dependent synchronization is very general. It's been seen from the level of single unit recordings all the way up to large-scale dynamics, like reconstructed sources with MEG and everything in between, depth electrodes, local field potentials, et cetera. Changes with aging, changes with development, and it has characteristic alterations which are associated with numerous neurological and neuropsychiatric populations. So it might be that going into the frequency domain, looking at oscillations, and looking at the temporal coordination of oscillations, the synchronization, how that changes in time, might be neurophysiologically quite important to understand network communication and its dynamics. So this is just an old slide, which is trying to essentially demonstrate visually Pascal Fraser's point. Whereas if you have two oscillating groups of neurons with a random phase difference, then the sending group of neurons action potentials arrive at a random time. Whereas if you have two synchronized oscillators, columns, Brodmann areas, et cetera, then the receiving area tends to receive input at the time when most of its neurons are at a depolarized phase, so that there's a maximal downstream impact. And that this is dynamic because even though the structural connections, as you see with DTI, don't really change on a quick timescale, certainly not the timescale of cognition, the expression of oscillations and the coordination can and do. So here's an example of how that would be is done in sort of modern network imaging using something like MEG, right? You'd have somebody sit in a scanner and then you'd say, okay, I'm gonna project the modeled broadband neurophysiological activity from 171 locations, something like a Brodmann area timescale. Then I'm gonna divide it using digital filtering into activity at these different frequencies that we know have different generators and functions. Inhibition in the case of alpha, active cognition and binding in the case of gamma. Then we're going to take its phase information or its amplitude correlation. We're going to look how that coordination is happening between areas. Then we're going to do a network analysis, like a simple standard graph analysis to see, are there hubs, are there clusters of brain areas that are working together? And then we can create a map for an individual person, for an individual moment, or for group averages. So this is an example of how that was used with post-traumatic stress disorder from military veterans. So this is showing hyperconnectivity in the gamma band. So that might be related to like hyperarousal, hypermemory, hypervigilance, this type of thing. And then you extract some graph properties like hubness. Is this a communication hub for that? And correlate it with clinical scores like patient health questionnaire, et cetera. And interestingly, this also shows how imaging in the frequency domain can be useful because, like I said before, if you have a traumatic brain injury or a lesion or previous surgery, you tend to get slowing of that alpha rhythm. And neurologists know that even in the critical care wards, they see it with those 20-channel EEGs. But that's quite different from PTSD. So even though the two conditions are hard to distinguish clinically, they're quite different in the frequency domain, which is why MEG is good. And I almost don't need this slide because of the last talk, but the point of this is now moving from a static to a dynamic point of view where people are doing exactly what I just talked about, except instead of doing it as a static, let's assume that that neurophysiological communication is going to be stable for 2 minutes or 10 minutes. Let's break it up into windows, right? Let's look at how that connectivity changes. And then can you say, does it change with age? You know, does it change with sex? Does it change with clinical group? And then you can, of course, couple that with modeling to make sort of predictions about whether or not your understanding of, to sort of try to validate whether your understanding of the underlying mechanisms of how those connectivity dynamics come about is true or not. So I'll end where I started. All tools are good, all tools are bad. They all give us something. They all constrain our knowledge, right? EEG is great, but it has bad spatial resolution. MEG is a little bit better, but it's still as bad at deep sources. It's still bad at radial oriented dipoles. FNIRS has bad depth. fMRI is fantastic. It has, for whole head non-invasive functional imaging, it has very high spatial resolution. It's good at deep sources. Doesn't matter what the orientation is, but it's an indirect measure and it's slow. It's not at the time scale that you're thinking and that I'm thinking. It's at a slower time scale. So take home messages. DTI is sort of our dominant way of getting the connectome. fMRI has been extremely prolific in informing the functional connections through blood oxygenation level dependent signaling. EEG and MEG have added the sort of direct measures that allow you to go into timing more accurately, frequency space. And now the focus is on how these functional connectivities are constrained, arise from and are constrained by the specifics of the underlying structural connectome or pattern of structural connectivity. And with that, I'll thank you. But in the interests of shameless self-promotion, I will have to say we're currently recruiting for a postdoc and maybe a PhD student or master's student in a project on neuroimaging, the basis of religious cognition and spiritual cognition, as well as several probably more technically oriented PhD students on combination of artificial intelligence, clinical EEG, and sort of patient natural language processing in clinical data with large sample 40,000. So Jobs. And Randy, do we have any question? Any time for questions?
Great, thanks. I've got time for a couple of questions. Go ahead, please, we're in the front.
I want to ask is maybe a little bit more in cognitive that, but when I hear about you talking about the sliding window kind of method, like any kind of method.
We're looking at this functional connectivity cycles kind of continues over time, and I know Dr. Jerse's talk that was also brought up. Do you think it could have applicability for not just looking at what the brain regions are lighting up in sync times, but also to do with what information is being encoded in these brain regions? That is to say, like representational similarity analysis being captured over time using a similar kind of sliding window methods.
So you're saying like if you identify there's a repeating pattern and then you look across brains and you see similar repeating patterns, can you sort of infer the cognitive content of that? Is that your question?
I'm asking more like not like within a specific brain region, kind of in a specific brain region. We look at the content of information in that brain region maybe in a similar way with a similar sliding window method, just kind of taking the sliding window methods and applying them to looking at information changes in a single brain region rather than shifts in connectivity.
Yeah, absolutely.
Yeah. Talk, it's going to be that question that in 15 minutes. So maybe you'll be able to answer it more completely after that.
Yeah, so I'll not get too specific an answer because I'm gonna have a 45-minute session on why I was so wrong. So yeah, I guess tune in for Rebecca Todd's talk.
But we would love to hear your take.
Oh, sure. I know you were talking about repeating functional connectivity patterns, and I was wondering if you're also interested in looking at that with EEG microstates, as it allows you to look at a higher temporal resolution. And see that they're like they're discrete and like non-random transitions between electric potentiometers.
Yeah, definitely. I would say that this has got to be related to EEG microstates.
Yeah.
We've done some stuff looking at maybe correlations between microstates and fMRI. And as a sort of in-between measure, I don't know, have you seen the work of Pasquale et al.? Essentially what they did is they created a method where they used a MEG and they sort of seeded the various resting state networks and then looked at amplitude envelope correlations and how those fluctuated. And then instead of, predefining a sliding window, like I'm going to go 10 seconds, 10 seconds, 10 seconds, they were actually able to identify by pulling out of the data, like default mode network dominant moments or salience network dominant moments, which is a bit like a microstate, but also more anchored in resting state networks as we understand them from an fMRI.
Yeah.
But yeah, I mean, I think this is highly related to EEG microstates.
One more good question.
Thank you for the presentation. I'd like to know if if we have, for instance, an information coming from different sources. So like we have different modalities of brain image. So I guess if you are capturing the information from these modalities, is there any way of how to combine these modalities to maybe
We do that. Yeah, definitely.
It's a good segue into my talk.
And as we have it, Brent's going to talk. Good.
Thanks. Thank you. Thanks again, Sam.